Okay, perfect. Well, good morning, everyone. Welcome to our March seminar. And we are very excited to have Mary Claire Veranso talk about her research today. Mary Claire did her undergraduate research and education in Cameroon, where she's also from. And then for her PhD, moved to Germany to work with Gudrun Kaderei at the University of Mainz, and did an impressive amount of research during her PhD work. A number of publications that she has put together is impressive, covering systematics and taxonomy of African Melastomateae. But also I've skimmed through her publication list. It's not only Melastomataceae, there are a few other families in there as well. So that's pretty awesome. That gives a very broad understanding of plant evolution in Africa. And Mary Claire now holds a postdoctoral position at the University of Munich, also in Germany. And she just told us that she's currently going for the habilitation, so to actually become a professor at the university. Yeah, so I guess we can all be excited for a lot of further great work from Mary Claire. She also just said, we have worked out Africa, continental Africa pretty well, or like she has, but Madagascar still is a mess. So yeah, I guess there's a lot more to learn from her. And with that, Mary Claire, please go ahead and yeah, take the floor.
Thank you, Agnes, for the kind introduction. So good morning, good afternoon, good evening to everyone. Today, I'm happy that we finally got to pick a date for this. We tried this for over a year now to get this done. So I will talk a little bit today about what I've been doing from my PhD, mostly on the taxonomy of African members of the subfamily Melastomatoideae, and how we have changed this classification over the last years. So this is just an outline on what I'll be talking about today in brief. So I would begin by saying that the African members of Melastomataceae, like the New World Melastomataceae, are restricted to tropical Africa. And here on this map, you can find them in this green patches mostly. And they consist of 675 species in 41 genera, divided into 2 subfamilies. So I will talk a little bit about the subfamily Olispioideae, though it's not my focus. I hope one day maybe Doug would give us more extensive talk on this subfamily. So they are terrestrial shrubs and canopy trees. They have berry fruit, which fuel large and globose seeds, and also large embryos. So these are really like the main diagnostic characters for this subfamily. And in Africa, there are 4 genera within this subfamily. So Memesilon with about 172 species, Lindenia with 9 species, Patandra 1 species, and Wannekia with 49 species. And now to the subfamily Melastomatoideae, my focal group, or my group of interest. So this is a cosmopolitan group, so meaning they are found everywhere. They have a diverse habit, and you can easily identify them by this having opposite leaves, although there is now one exception, which we would talk about later. And they also have this typical acrodomous leaf venation. So you'll find, as you can see here, the flowers are diplostaminous, and they all also have poricida anthers. Within this subfamily, there are 2 validly published tribes, Melastomateae and the Sonerileae. Felicia ademia for this talk, I would so Felicia ademi, I would use it like this, but this is really not validly published. And also talk about the Unplaced tribe. So I begin with the Unplaced tribe, and this Unplaced tribe consists of two genera, so Dinophora and Ochthocharis. Dinophora is endemic to Africa. Ochthocharis is disjunct between Africa and Southeast Asia. And these two genera, you can easily identify them as terrestrial shrubs. They have this long and weak petioles. Their leaves are their leaf margins are serratidentate, and the anthers have a dorsal spore. Maybe I talk a little bit about Dinophora here because it was kind of interesting to me somehow because it had been placed in Gravesia before, also placed in Melastomataceae, and also placed in Dissotis. But when we sequenced this Dinophora, it was not in any of these tribes. So we later included sampled Ochthocharis, and this Ochthocharis dissolandoides is from Africa and bonensis from Southeast Asia, and these three form a monophyletic clade, not sister to, so not within Dissotis or Melastomataceae or Gravesia. And we think this probably should be treated as a segregate tribe on its own, which I think Darren is already working on this and would publish this probably in the Melastomataceae book, which will be published soon. So we would have a new tribe for this two genera. And here are just some pictures to see how this to see these two genera in Africa. Then we have this, I go to talk about the Phylogeny. So it's a monogeneric tribe with only one species, Phylogeny stenocarpa. And you can easily identify this species by their keel calyx lobes. They have dimorphic stamens. The anticephalous stamens have frustrate anthers, and the ovary is inferior, and it's endemic to Guinea. This species is only known from the type collection, which was collected in 1949. However, recently Q did an expedition to Guinea, and it has again been collected. That's why I have this picture. So I have already extracted the DNA, and which is quite good. So I think we'll be able to, in the next months, place this species to know exactly where it falls within the family. And I go talk about Melastomataceae, which it's going to be like most of the talking today. It's really based on this, the tribe Melastomataceae. Worldwide, there are about 823 species in 44 genera. The African-Malagasy group, so this here, consists of about 193 species in 25 genera. And we can easily identify Melastomataceae by their uniporous anthers. The stamens have pedoconnectives and bifurcated dorsal appendages. The ovaries are half inferior to superior, and they always have this persistent crown bristles or trichomes, which if you try to look a little bit in this picture, you would find here or here. The fruits are capsular, most of them are capsular or capsules, with berries not so common, for example, in Melastoma. The seeds are cochlate with tubercles, as you can see above. So within the African members of Melastomataceae, there is a huge morphological diversity. For example, we have flowers, which are mostly pentamerous and tetramerous. However, we also have few exceptions of even up to 7 merous flowers. Color is also very variable. We have yellow flowers, white, pink, purple, and several things in between. Fruit type, most of them are capsules, except, so I'm talking about the African members, except for Tristema, which have these fleshy fruits here. Inflorescence, we can have solitary flowers, cymes, or one to few flowers, or even panicles. And one of the most diverse characters within the African Melastomateae is the hypanthium or calyxtic pubescens, for ranging from glabrous to simple hairs, then stellate hairs, or a mixture of both. Or for example, here in Tristema again, where we have just this ring, or this, we have this ring of crystals, but generally glabrous. Or here in Adurella, where we have dense stellate hairs mixed with glandular hairs. I talk a little bit about staminal character here because this is very important within the African Melastomateae. So we have the main two types. We have maybe variations in between, so sub-equal, but the two extremes would be isomorphic stamens, where we have just one, the stamen types are same size, same shape, color, or dimorphic, where they vary in size, in shape, and sometimes even in function. And why this stamen character, it's very important. It's because Osbachia was thought to be only to only have species with isomorphic stamens, and Dissotis with dimorphic stamens. So because of this, there were lots of transfer from between these two genera. We will come back to stamen morphology, or how important these characters are, in the next slide. So during my PhD, one of the main objectives was to test monophyly of all the currently recognized African Melastomateae genera. And for this, we use ITS and two plastid spacer, ACCD PSI and PSBK TSBL. And the reason for this was because we wanted to use as many outgroup samples from the New World, which had been published some few years back by Fabian and others. And these are the results we had from this phylogeny. So what you can see in blue are all the species which were treated in Dissotis. In green, the species which were treated in Euheterotis. In brown, the species in Osbachia. So what we can already just looking at this just tells us that Dissotis, Euheterotis, and Antheratoma, these are all polyphyletic, Osbachia or polyphyletic genera. And we mapped, manually mapped morphology stamen type. So here, and here you see that this is not really a good character because this is quite variable and not stable. So designating a genus just based only on stamen type shouldn't be something shouldn't even be within the whole concept. Maybe with other characters, it's fine, but we can't just designate genera or genus based on only these characters because they are really very not stable. The next slides, I would maybe zoom in a bit into the different clades. So the first clade is this pseudoheterotis clade. I call it the pseudoheterotis clade because we have two sections. So heterotis section Adurella and heterotis section Cyclostema, which were all part of Euheterotis, but Euheterotis and Stricto were now excluded. That's why I just call this clade Pseudoheterotis clade. So it's an informal name. So the first lineage we covered here, it's the Guionia clade, which now consists of Guionia plus all the species previously treated in Heterosis section Cyclostemma. The next lineage is the Euheterotis pubicina lineage, which we described a monospecific genus because the species is really morphologically distinct and it doesn't fit to any of these two clades. The next lineage is the Adurella lineage, which composed of all species previously treated in Heterosis section Adurella. And then we have the Melastomataceae lineage, plus one Osbachia species, Osbachia potterici. So Osbachia potterici, let me talk a little bit about Osbachia potterici. It's the only species within this genus which has isomorphic stamens. However, excluding isomorphic stamens, the other characters fit well to this. It makes sense to place it within Melastomataceae, which was, I don't know, because they used this anti-character, that's why I think this was excluded. So saying stamen type was isomorphic, and it moved. Tristema was one of the few clades we recovered, which was well supported within the African Melastomateae. And this makes a lot of sense because most of the species are really good morphologically because of this ring of bristles surrounding the on the hypantheum. So I just put some pictures here to talk about how these different genera can be identified. So the first one is the Melastomataceae, which consists of 5 species, and most of them, they have mostly solitary flowers or few flowers, surrounded or subtended by this involucral bracts, and they are dimorphic. Tristema, on the other hand, have these fleshy fruits. They have isomorphic stamens, and what is very diagnostic, it's this ring of bristles on the hypantheum. Adurella consists of 7 species, mostly found in marshes, and they have the hypantheum is covered with dense stellate and glandular hairs, and they also have isomorphic stamens. Guionia are small herbs, so mostly from small herbs rooting, not always, but most of them root at the nodes. And they are also geophytes. The hypantheum is glabrous or rarely with simple hair. So the next clade is the large Euheterotis clade. I will talk about further divided them into 2 clades. So Euheterotis clade 1, which I'm going to talk a little bit now about. So the first lineage we recovered here, it's the Decanthathera lineage. And in this clade, all the African and Madagascar members sampled are recovered in this clade, so supporting monophyly of the genus. Dissotis consists of all the species previously treated in Dissotis subgenus Dissotis, while Euheterotis now here, it's really Euheterotis and Stricto. The next lineage, this lineage here, it's the Asian Melastomataceae lineage and consists of 1 clade of Osbachia and Melastoma. And interestingly, this clade, the type has nothing to do with, of Osbachia, has nothing to do with the other African Osbachia species sampled. The next lineage, it's the Madagascar endemic lineage. So Amphiblemma, which is here, Dionysia, However, within this clade, there is really no support. And it would really be nice to densely sample within this clade to see what exactly is happening here. The next lineage is the Dupinata clade, which is composed of all the species previously treated in Dissotis subgenus Dupinata. And last lineage here is the Pseudo-Ospeikia lineage, which is a monospecific genus. So again, we can see here that some pictures to differentiate or help us identify these different genera. So Decanthanthera here has 8 species in Africa. There are 28 species in Madagascar. And these are all herbs or shrubs or trees with pentamerous flowers. They have coarse leaves and dimorphic stamens. Disotidendron, on the other hand, are shrubs which are deciduous leaves. They are pentamerous and they are endemic to East Africa, with only 1 species extending to Democratic Republic of Congo. Euheterotis sensu stricto now consists of 6 species, and these are all decumbent herbs with mostly these rhomboid leaves. Their hypanthium has stellate hairs, with the exception of 1 species which has simple hairs, and they also have dimorphic stamens. Dupinata has 5 species, and these are all herbs or shrubs, and they have pentamerous flowers, dimorphic stamens, and they are the anticephalous appendages have tricuspided. And one other very important character distinguishing Dupinata from the other genera are this acrescent capsules or acrescent fruits with this longitudinal slits. And the last clade is the Euheterotis clade 2. And this clade, or also called Dissotis and allies clade, consists of Dissotis, subgenus Dissotis, with all its 4 sections, Anthraeotoma, and the rest of the African Ochthocharis species. I would come back to this Euheterotis, Dissotis and allies clade later on, because here the support wasn't really good. So we didn't propose any taxonomic revision at this time for this clade. The next genus I will talk about is Notodissotis, and this is a new genus we described from Atlantic Central Africa, and it consists of 2 species, Notodissotis alinensis, which is restricted to Mount Allen in Equatorial Guinea, and Notodissotis battery, which is found in southern Cameroon, Principe, and also the border between Equatorial Guinea and Gabon. And this genus consists of shrubs with deciduous leaves. The calyx lobes are notched, so you can see here, and they are asymmetrical and caducous. They also have this stellate hairs or tuft of hairs on the apex of the calyx lobes, and the stamens are dimorphic. Morphologically, it was easy to recognize when I saw Notodissotis alinensis that this was a new species, but I wasn't sure which genus to place it. So we were only able to describe this like a new genus only after we did were able to sequence this. So it was recovered within Melastomatae, which makes sense because they have this cochlate seeds with this tuberculate surfaces. The next genus I talk about is Chylila, and this is also endemic to Guinea. It was only known from its type species from 1937, but again, it was recently collected from Guinea, and we were able to sequence this species. So if you look at this map, you would find that now Chylila, it's not, so there are several populations of Chylila all in Guinea. So at least there are more collections available now for this species. So it's a monospecific genus with a March branch deciduous shrub, so you can see here. The juvenile plants have a paterculus base. The flowers are terminal, solitary, and subtended by an involucre of leafy bracts. The stamens are isomorphic, the large pediconnective, and the dorsal spore is present but very short. The ventral appendages are linear. Again, here we see that the seeds are cochlate and also tubercles on the surface, again supporting that it should be in Melastomataceae. And this was also supported by the phylogenetic placement of the species. Now I talk a little bit, go back to Dissotis and allies. So here you can see how diverse this Dissotis and allies clade is. So from flower type, shape of petals, number of flowers, hypanthium pubescens, habit. So what happened was we decided to sequence additionally ETS and also NDHF to see if we could better resolve relationships within the Dissotis and allies clade. And yeah, we were able to do this, and it was better resolved, and we recovered most of the clades which were morphologically proposed for Dissotis. So the first clade we recovered is the Rosetia clade, which consists mainly of species which were mostly treated within this section Squabulosae. The next lineage was the Rosetia lineage, consisting of this monospecific species, so Dissotis tubulosa. The next lineage is the Dissotis sensu-stricto lineage. So this is where we have the type of Dissotis grandifolia. And then the next lineage is the Neurophila sensulato lineage. And this is quite interesting because Neurophila, the one in orange, was transferred to Euheterotis, a new world genus. But when we sequence this, we support that it has no relation to the new world Melastomatae, but it's really an old world genus. However, several other species are recovered in this Neurophila clade. So we have in yellow here, 2 Osbechia species, and in blue here, 2 Anteretoma species and 1 Dissotis species in this clade. The next lineage we recovered is the Anteretoma lineage, which is more or less a good group if we exclude this 2 species. And then we have 1 monospecific lineage again, so the Almedanthus lineage for Dissotis pachytricha, which is also morphologically unique. The next lineage is the Euheterotis lineage for Dissotis section sessilifolia, and another monospecific genus for Dissotis gilgania, and then the large clade of Phylogenotis, which consists of all the species previously treated in Dissotis macrocarpine. So we did a morphological character mapping to check if we should treat Dissotis as one genus, so a broad treatment of Dissotis, or to split Dissotis into several genera. And what happened here, as you can see, is that we have no characters which unite all of the species here. So if we treat Dissotis as enlarged, then we would just be where we are again today. So it just becomes again this, maybe dustbin genus where everything you're not sure of would probably just end up coming back to Dissotis. However, these different clades are morphologically very distinct. Some of them geography also contributes, habit, so we decided to split this Dissotis. I'll pick some few genera within this clade, and then we can just discuss about how we can morphologically identify them and how distinct they are. So the first one is the Dissotis sensostricto clade, this here. It consists of 6 species, and these are all herbs. They are very hairy, and their hypantium is covered with stellate hairs, stipicate stellate hairs, and their sepals are glabrous, except for the apex, which is stellate, or you have a tuft of hairs, and they are generally dimorphic. The Roseifia is one here, are annual herbs, and they are pentamerous or hexamerous. And what is very diagnostic, it's this fruit where you have the base, it's kind of subspherical with persistent stellate hairs, and the above or the apex of this fruit, it's oblong, so cylindrical with simple hairs. And now the enlarged Neurophilia clade. We have species which are either pentamerous or tetramerous, and they are all herbs. They are hypantium are ovoid, and they have simple hairs. Anthractoma, on the other hand, are strictly tetramerous, and the flowers are they have persistent bracts, which are very tiny, and they also have these very distinct intersepal appendages. Phylogenotis, this last clade here, consists of 12 species, and they are also shrubs, most found in open habitats or grasslands, and they have this large bracts. The hypantium, it has this silvery to serraceous hairs. And Euheterotis, this right here, has 4 species, which are all found in marshes or swamps, and they are mostly glabrous, so both the hypantium and the entire plant, and rarely, or some, except one Glaberrima, where you have this rare sparse hairs. So the next clade I talk about is the Ammedanthus, the genus Ammedanthus, and these are subshrubs with glabrous leaf or sparsely oppressed hairs. The leaves are purpuraceous or discolorous, and they are pentamerous. Flowers are pentamerous, and usually one or few. The hypanthium has this large reflex appendages, which I don't know if you can see here. And these appendages are probably end with 1 to several setae. There are actually varieties within the species, and these are all differentiated based on how many setae are found on this scale-like appendages. The next genus, it's the Pyrotis, and this is a low perennial herb with numerous stems arising from a woody underground rootstock and the 4 tuber-like structures. When you look at herbarium samples of this species, they always appeared burned, which indicates that probably these tubers are formed when in adverse conditions, and then again resprout when conditions are favorable. So probably fire, it's one of these adverse conditions. And they also have this attenuate leaf bases, which end up forming like contracted petioles, or sometimes you can't even distinguish them. And their inflorescence is a cyme. There are 2 varieties in this for the species, and all of them are the 2 are distinguished mainly based on the petiole and leaf shape. So to summarize what we the taxonomic decisions we propose for this tribe Melastomatae, we reinstated 4 genera, so Adurella, Derosifia, Dupinate, and Derophila. And we propose a new status for Dissotis subgenus Dissotidendron and broaden the circumscription of Guiaumia. 7 new genera were described: Almedanthus, Euheterotis, Eleutis, Phylogiotis, Dissotis, Pyrotis, and Rosettae, and a narrow circumscription of Dissotis sensu stricto and Euheterotis proposed. To complete this generic realignment, 12 synonyms were proposed, and a total of 101 combinations, and 57 lecto or neotypes were also designated. And now we have a revised key for all recognized African Melastomateae genera. However, 2 species are still unplaced, so Dissotis lunense, which is endemic to Guinea and Sierra Leone, and it's a morphologically unique species currently treated in a subgenus Dissotis, so subgenus Paleodissotis, and the lone species in the subgenus. We also have Dissotis splendens, which is also unique, and currently I can place any of these 2 species in any of the recognized genera. Currently, we have samples for this Dissotis lunense, which was again recently collected from by a Q, and we should be able to have placed this in the near future. So I will talk a little bit about Sonerileae. Most of the work discussed here is not published, but the manuscript is submitted. So worldwide, we have approximately 1,080 species in 44 genera, and the African-Malagasy species are about 240 species in 8 genera. This is a very diverse tribe in terms of habit, inflorescence. I don't know, it's really like, it's one extreme tribe to work in. So we have fruits, which are berries and capsules. Capsules are very variable, and the seeds are numerous and with diverse shapes. The African members of this tribe consist of 56 species in 8 genera, And they are mostly restricted in the Central African rainforest here. Cincinnabutres and Gravesia have their center of endemism, probably here in East Africa, in Tanzania. And Benna and Buderia are endemic to Guinea. Madagascar seems to be the center of endemism and hotspot for two genera, so Gravesia and Medinilla, with 111 and 73 species respectively. I talked briefly about Benna. So Benna is a new genus which we recently published about 2 weeks ago, and it's a new herbaceous genus from Guinea. You can see here from the illustration. So you find them in these canyons and these vertical cliffs, and it has alternate leaves, which is quite rare within the family. We were able to sequence the species and placing it. It's within the neotropical endemic genera of Sonerileae. So weakly supported assistance to Finantia here, and not related to the rest of the African Sonerileae members. And now the phylogeny of African Sonerileae itself. So we have this here. We sampled one African Medinilla mani, which is recovered within this largely Asian clade, where other Medinilla species are recovered. However, the backbone for this clade is not supported, so we don't really know what is happening within this clade. The African Madagascar and the other endemic genera, so Gravesia and the rest, form this well-supported clade. And Gravesia, it's well-supported, although it's still very poorly sampled. And Cincinnabutres, it's not monophyletic, because one species, Cincinnabutres felicis, is recovered out of the Cincinnabutres sensostrictus clade. And Amphiblemma is also not monophyletic, because Amphiblemma molle is recovered out of the Amphiblemma sensostrictus clade. So here, this is the African Medinilla we sampled. So Medinilla mani here, which seem to be sister or nested with some Madagascar Medinilla species sampled. However, one species is still here. And then in blue here, you can see the other African, where the other African Medinilla species are placed. But like I said, this backbone is not supported, so we can really rely on this. And these are just some pictures to show you how diversity within Gravesia, like Medinilla, Gravesia is also a super diverse genus. But the phylogeny supports that the African members are sister to the rest of the Madagascar Gravesia species. But yeah, we still need a lot of Gravesia samples from Madagascar to test the infrageneric classification within this group. These are just more pictures of Gravesia from Madagascar. The next clade I talk about is this Buderia felixi clade and the Cincinnabutres clade. So Buderia felixi was once before treated as a monospecific genus, but was later transferred to Cincinnabutres as Cincinnabutres felixi. But recently, QA again collected the species from Guinea, and I was able to sequence it, and it falls out of Cincinnabutres. And here you can see that it clearly differs from Cincinnabutres sensostrictus. So Cincinnabutres sensostrictus are caulos herbs with the flowers arising from the base, the lack internodes. However, in Budaria or Cincinnobotrytis felici, they have this comos base, very short internodes, and the leaves are not always arising from the base because they have nodes. And the flowers are born at the top axillary, so at the end of the where the leaves end. The next genus is Dissolanthera, which is also shrubs in tropical rainforests, well supported by both molecular and phylogenetic analysis. Then we have Cavoa, which are easily identified by this petals, which are asymmetric. And then the calyx and the ovary are adnate, and they form this woody wedge-like excentric crown. And the flowers are always scorpioid, and the inflorescence are scorpioid. And here we have the Amphiblemma sensu-stricto clade, so this one. And these are mostly herbs. And they are covered with dense hairs, most on the Flora parts. While Amphiblemma molle has this typical cincinnate uniparous inflorescence, which is different from the rest of this Amphiblemma species. So this is how a typical Amphiblemma molle would look like. And the last genus I talk about, it's Procelia. And this is these are mostly epiphytes in rainforest, and they are pentamerous. They have this ellipsoid fruits, which are not so, the angles are not really prominent, and they have isomorphic stamens. And yeah, this is, I just wanted to maybe give you like an overview of where we are with the African members of the subfamily Melastomatoideae. And with this, I would like to acknowledge Professor Gudrun Cartwright, Dr. Stone, and Hannah Mendes for she was a bachelor student who did part of the work for the Sonerileae, and Zanda for collaborating with us and providing us these very important samples from Q, and the lab as technical assistants, and all the group members from the Gudrun and Joachim Cartwright working group. And curators from different herbarium have also been very supportive with providing us with samples. And to everyone who is not mentioned, who somehow supported in this work. And funding was gotten from University of Mainz, IPT, ITY, DAD, and DFK for this work. So thank you for your kind attention.
Thank you so much. This was a great talk, Marie Claire. Are there any questions? I'm sure Doug has some.
Yeah.
So I have one. I will start. So Marie Claire, in most group of melastomes, when they are in different continents, we've seen that it seems to be still very structured. So like in Melastomataceae, sorry, they are in the neotropics, then they go to Africa, and then they go to Asia or something like that. But in Sonerileae, it seems that it's not quite that easy. It seems that there's been multiple east to west and west to east dispersals. Do you have any thoughts on that? And I know the sampling is very preliminary, but any thoughts?
I think the only thing I'm thinking about is Guinea. When this question is asked, it's really Guinea, because even when you look at Melastomatae, the oldest, so the old lineages are always this restricted, most of them are restricted to Guinea. So it seems like Guinea was kind of like this cradle of Melastomataceae before moving to other parts of Africa. That's the only thing I can say, but really what's happening in Sonerileae, I really don't have a clue. Yeah, it's really strange. It was a strange result we got there.
I have another question. I saw in some of your pictures in the background that some of the species apparently grow in wet areas. So I wanted to know whether there's any sort of habitat association that you see, like specific clades, tribes, sorry, not tribes, but like, yeah, lineages occurring mostly in whatever specific habitat types, or if there's no habitat structuring at all.
Some of them are really restricted to certain habitats. For example, Sonerileae is restricted to tropical rainforest, so primary forest. You don't find anything in open habitats. They are really restricted to tropical primary forests. But with Melastomatae, some species are restricted to closed habitats, some to open grasslands, and within the genera, some are restricted to maybe marshes, or so you have this kind of there is a bit of structure, and also at the same time there is no structure for some genera.
Yeah, like no easy habitat association that would help in the systematic placement, I guess.
Some are well structured, but not all of them.
Bravo, Marie Claire.
Yeah. Thanks.
What about Dionicastrum? It's from Tanzania. Is that another paleo-endemic, do you think?
That's a good question. I don't really know about what is going to happen with that one, unless I'm able to sequence it. I think it's still a mystery about, I really don't know what I would say. I have no clue where to place it. So I'm not sure if I would treat it like a paleo endemic genus or something. I don't know.
So it's still very mysterious.
Yeah, to me, it's still very mysterious.
Okay. I'm also interested in what you and maybe others think about all these shifts between staminal isomorphism and dimorphism in the Melastomatae, and what could be driving that, or how could we learn about what's driving that?
I thought of maybe, when I started, I thought maybe this was like a pollination driven, this had to do with pollination or pollinators. But then I realized that my personal experience is that maybe flower size also matters in this. So I think flower size, because when we tend to have smaller flowers, we tend to have isomorphic stamens. But apart from that, I can't really tell you much about what exactly is happening with why we have this lots of shifts. Maybe, Fabian wants to answer.
No, I agree that on the flowers, but a question to you is that, and maybe, I don't know if you have looked into this, is when in one of your papers, earlier papers, you looked at the transition between open habitats and forest habitats. And I wonder if there is any correlation between the open habitat in heteroanthery and closed habitats and isomorphic anthers.
No.
Okay.
It had no relation.
And do species with either anther type usually co-occur? So isomorphic and dimorphic?
No. Actually there is no species with having both types of anthers that we know of.
No, but I mean, do species with isomorphic anthers occur in the same habitats as species with dimorphic anthers?
Yeah.
Okay. Yeah. So
Yes.
there's no, because otherwise you could have assumed there's maybe some major pollination biological structuring, but maybe
No,
it's
they do occur.
the pollinators better.
They do occur together.
Yeah. More work to do.
Yeah.
Any other questions? Well, it looks like everyone is mostly happy.
Very interesting. I'm still impressed by Benna. That plant was amazing. And I was impressed how similar it looks to this weird thing from the base of the Andes, Kipuanthus, even though it comes out in such a different group. It's cool plant, cool discovery. Congratulations on that. And not surprising that comes in a weird place.
Thank you.
Okay, well, I will stop the recording.